Hi everyone, welcome to Doc Hill's study where I help you get ready for your National 5 biology assessments. In this video we'll be walking through questions from an official SQA paper. The paper and the marking guide are both produced by the Scottish Qualifications Authority, the SQA, and all rights to that material belong to them. I'm using these resources strictly for educational purposes here on this non-commercial channel to support students like you in preparing for your exams. I'll be using the SQA's marking guide to highlight what answers are accepted, but keep in mind it usually only shows some of the correct answers and doesn't explain the reasoning behind them. That's where I come in. For each question, I'll walk you through the thinking process, explain what the question is really asking, and point out some of the common pitfalls to avoid. If anything doesn't make sense to you, or you want to ask about a specific point, feel free to leave a comment. I'll do my best to help you out. If you find this helpful, please give the video a like and subscribe to the channel so you don't miss any future walkthroughs. Let's get started. We've got a bit of work to do here by the looks of things. An experiment was carried out to investigate the effect of carbon dioxide concentration on the rate of photosynthesis. The rate of photosynthesis was measured by recording the mass of sugar produced per hour. Okay, there's the mass of sugar produced per hour. Grams per hour, carbon dioxide concentration in percents. Let's see if we can figure out the graph. Well, it doesn't start at 0, so make sure you note that just in case. And we go for carbon dioxide concentration has to get to a certain level before it picks up. And then as carbon dioxide increases from We'll figure that out in a minute from there, but we'll figure that out. It's going to take us a bit of thinking. As carbon dioxide increases from there, it reaches 0.08% concentration and then levels off. Okay, so it looks like extra carbon dioxide is not causing any more increase in the amount of sugar production. There must be some other limiting factor. We've got a line here that says X. Okay. Describe the relationship shown between carbon dioxide concentration and the mass of the sugar produced. Describe. Okay. This one is a wee bit harder. You can't just say the independent variable, as the independent variable increases, the dependent variable increases as well, because here's the problem. For starters, the carbon dioxide increases. Okay, it increases from a point that isn't quite 0, but we might get away with that because there's not very much in it. And we can say it increases, the mass of the sugar produced increases until, and we have to finish this off or else we're not going to get both marks, until the carbon dioxide concentration reaches 0.08%. And then it levels off. So that's what we're going to write, okay? So what we can't just do is say as carbon dioxide increases, the mass of sugar increases, the mass of sugar produced increases, because it levels off. If we're going to be really strict, we might have to say as it increases beyond, what is this? That's 0.04, so that's 0.02. I don't think they're going to ask that, so let's not bother about it. It's only 2 marks anyway. So let's say let's go independent variable, as the carbon dioxide concentration, write out the whole variable. Okay, don't just write as the carbon dioxide. As the carbon dioxide concentration increases, not a lot of space for this one actually, it's quite a lot here. The mass of sugar produced, they're saying here you don't have to put the mass of in, produced, but it's safer to do so. Just write out the whole variable that's on the label. Produced increases. And then you can say before leveling off at 0.08% CO2 concentration. You're not allowed to write in there, right? So you've got to make that, you might have to put it underneath. Okay, that's quite a lot to fit into those 3 lines. Your writing's probably smaller than mine and better. So you've got to say, then levels off. If you don't say levels off, you don't get the second mark, okay? You get 1 mark for this, that's the relationship there. You get 1 mark for the leveling off. It's a wee bit funky there that you don't start it absolutely at 0. Anyway, let's move on. A lot of students will have found that one difficult to get, I think, that mark. Let's have a look, 13A1. Yes. Yeah, so they didn't say it leveled off, so you didn't get the final mark. A lot of students didn't do that. Okay, make sure you look at this. If you see 2 marks, there's 2 things you're going to have to do, so look for the 2 things. There's 3 marks there, I would have had to deal with this, I think. But the 2 obvious ones are this and this. And this. You probably didn't see that. If it was 3 marks here, I might have had to deal with this, but just because it was 2 marks, I'm just dealing with this one and this one, okay? You have to be a little bit, thinking about what the examiners are looking for. All right, so just one factor that could be limiting the rate of photosynthesis at point X on the graph. Well, we could put one of the other limiting factors of photosynthesis. What are the limiting factors of photosynthesis? Well, they could be temperature or they could be light intensity, right? Temperature eventually is going to slow down your reaction because at some point the temperature is going to be a limiting factor. So in this case, temperature is going to be acceptable. What you can't write is just light because it doesn't suggest a measure of it. I'm going to stick to the safe one for photosynthesis. You should always stick to the safe one for photosynthesis. The other limiting factor for the rate of photosynthesis is light intensity, carbon dioxide concentration and light intensity. Okay, because it's governed by enzymes, temperature is a factor, but I wouldn't put it, you'll get the mark, I think, for temperature, and you do get the mark for temperature, but I'm going to stick with light intensity because it's very specific to photosynthesis. Okay, so that was question 13B2. Oh no, that's okay, B1. Okay, let's have a look. Hydrogen is produced as a product of the light reactions and is required for carbon fixation. Describe how this hydrogen is produced. Well, it's produced because water molecules are split by the energy absorbed by chlorophyll in the chloroplast. Okay, so we're only asked 1 mark here. Hydrogen is produced by splitting water molecules. Okay, that's rather deep knowledge. There's a lot of students who won't have got that, I think. Okay. You don't necessarily, as a biologist, you probably don't need to say water molecules by splitting water. Remember, H2O, water is H2O. The oxygen is the waste product, so the hydrogen is kept, the oxygen is dumped, and that's what we call splitting a water molecule. If you say that in some other way, like the hydrogen comes from water and the oxygen is exported as waste or whatever, you might get away with that as well. You just got to be careful. Splitting water molecules is the way we generally talk about it. So we split the water, we keep the hydrogen, we dump the oxygen. The oxygen just diffuses out of the plant into the air and keeps you and me alive. Okay, the sugar produced during carbon fixation. The sugar produced during carbon fixation stage can be converted into other substances such as starch. Okay, name one other substance sugar can be converted into and state its role in the cell. Okay, well, the obvious one here is one of the, we've got the plant polymer, which is starch, one that's the storage polymer, the polysaccharide as we call it. So but polymer, or you can think of it as long chains of sugar molecules. In this case, the substance we're going to think of is cellulose. That is converted from the sugar into cellulose, and the role is the cell wall structure. Or the structure of cell walls, okay? component of cell wall structure. Now I think that there's possibly another answer here, because if you remember your respiration, the sugar produced by photosynthesis is broken down to pyruvate, so it's converted to pyruvate in order to produce ATP in the first stage of respiration. You might get away with that, it's not in the answer sheet, but it's not wrong as far as I can think of, so you might get the mark for that. And the role of, you'd have to say what the role of pyruvate was, and pyruvate is the raw material for aerobic respiration in mitochondria. So that would be a much more difficult one to answer, because the role has to be of the molecule, right, the new substance. Not, you can't just say into pyruvate and then to produce ATP, because that's what its function is, not what its not what the role of the pyruvate itself is, you know. So this is the function of the process, not the role of the pyruvate. Pyruvate's the raw material for. Anyway, I wouldn't go there. Stick to cellulose, easier. Okay. Thanks for watching this walkthrough from Doc Hill Study. I hope it helped you feel more confident about tackling these kinds of questions. Remember, the answers I've used are based on the SQA marking guide, but it's always important to understand the reasoning behind them, not just memorize the words. That's why I focus on the process, not just the final answer. If you've got any questions, or if something didn't quite click, drop a comment below and I'll get back to you. If you found this helpful, give it a like, and don't forget to subscribe so you'll be notified when the next walkthrough goes up. Good luck with your revision, and I'll see you in the next one.